Hello, my name is Jordi Rodó and I work as iLite researcher at Svar Life Science in Sweden. Let's talk about the assay development within the field of AAV therapies, engineering iLite cell lines for specific and analytical applications. First, let me start talking briefly about AAV vectors. As you may know, AAV vectors are replicative effective vectors that derive from human non-pathogenic parvoviruses. These vectors can transduce both dividing and quiescent cells and can mediate a long-term expression in non-dividing cells. Moreover, there's a wide variety of serotypes and we can manufacture high-titer preps. One of its mainly drawbacks is its small clone capacity, which is around 4.7 kilobases. Despite the impressive results that it has been obtained using AAV vectors to treat different disorders, the development and use of these therapies is normally combined by important challenges and hurdles. Therefore, the development of new tools that could overcome these challenges is a necessary step to improve the shift from preclinical stages into successful clinical studies. The development of relevant bioassays based on human in vitro cell lines are a valuable tool that can provide answers and mitigate some of these challenges. At Svar, which means answer in Swedish, the development of bioassays has been a very central part of our activities. We have developed the iLite technology, which are cleverly engineered cell-based representative system that not only can determine the binding interaction, but the functionality of the compound. Later on, we have been starting development bioassays for gene therapy, as our cells normally reflect the mechanism of action of the vector. Moreover, the iLite technology allows us to design and customize the cell line for any vector, target, or therapy. We commonly pick up a certain signaling pathway and we hijack this pathway to signal into the specific expression of firefly luciferase. The combination of chimeric transcription factors with synthetic promoters ensure a high pathway specificity. Moreover, our cells are provided in a zone-use format that not only decreases timelines and workloads, but gives superior performance. As depicted here, you take the frozen cells, you thaw the cells, and then you pipette these cells together with your vectors or your compounds. Then you just incubate this combination for some hours, and normally in the same day you have your results. So let's focus on the development of bioassays for cell and gene therapy, which highly depend on the outcome we want to assess. Therefore, first I will talk about the development of bioassays for specific therapies or vectors. Probably, if you're aiming to use a specific vector or for treating a specific therapy, you want to have a bioassay with a very specific cell type, because your goal is to transduce very specific cells, or you're using a specific promoter that could only be expressed in specific cell lines. Moreover, developing a bioassay that mimics the mode of action of your vector is also essential, as then you will have more valuable data. Therefore, this type of assay can be used, for example, for therapy optimization, for product similarity, or for determining the potency of the vector. Let me exemplify this by talking about the development of a bioassay for a gene therapy product targeting the eye. As you may know, the gene therapy for the eye is one of the most boosted clinical development areas, as the eye presents important advantages to be treated using gene therapy, because it's an immune privileged organ that is contained by physical barriers that prevent vector from disseminating systemically. And moreover, retinal cells do not proliferate, meaning that we can achieve long-term expression. It's a small size, so we don't need a large vector production, and we have plenty of animal models and technologies to study changes in this organ. Several eye disorders, such as retinosis pigmentaria or Leber congenital amaurosis, presents variations in the cyclic GMP metabolism due to mutations in the photoreceptor phosphodiesterase, which mediates the degradation of cyclic GMP, or guanylate cyclase, which affects the generation of cyclic GMP. Based on this, we have developed the iLite cGMP cell line to facilitate the development of therapies for these diseases. as we're talking diseases that are eye-related, we have used human retinal cells as a cell type for this bioassay, and moreover, our bioassay mimics the mode of action to have a more relevant bioassay, and our cells are provided in a throw and use format. Overall, you can use this kind of cells to improve your therapy, to assess product similarity, or to determine the potency of the vector. To run this type of assay, you just need to incubate the iLite cGMP cells together with your vector for 70 to 8 hours, and then you just need to determine the luminescence. As said, one of the potential applications of this kind of bioassay is to assess the vector potency. Here we have determined the potency of AAV2 and AAV5 by comparing a 100% dose response curve for both serotypes with a 150 and 50% dose response curve, also for both AAV2 and AAV5. Then we can assess the accuracy of this bioassay, which is high. This kind of results can help you compare the different manufacturing methods, compare the potency between different lots, etc. But as said before, the development of bioassays highly depend on the outcome you want to assess. So we could aim to develop a bioassay for a more analytical and characterizing application. This type of bioassay, for example, can help us to determine impurities, to determine maybe the full versus empty capsid ratio, or to assess the presence of neutralizing antibodies. %90 of the human population will be exposed to wild-type AAVs during his life, quite often during their childhood. In 2010, a French group collected hundreds of samples from human serum and determines the presence of neutralizing antibodies using an in vitro assay. This group judged a sample to be positive for neutralizing antibodies when a 1 to 20 diluted serum could inhibit more than 50% AAV transduction. The results from this experiment show that both AAV1 and AAV2 present a seropositivity among the population of more than 50%, while AAV5 and AAV8 present the lowest. Moreover, they observe a correlation between the percentage of seropositivity and how strong was the titer in this population. As NAbs against AAVs are commonly found in the population, each assessment is beneficial and required before, during, and after the treatment with AAV-mediated gene therapy. Let me show you how can we develop a bioassay for determining the presence of neutralizing antibodies by using as example the iLite AAV neutralizing platform against AAV of serotype 2. However, this system, I have to say, is quite modular, so it can be adapted to other serotypes. This system is composed of a two-cell setup. First, as you can see in your left-hand side, we have the AAV2 packaging cell line that has the ability to produce AAV vectors, and on your right-hand side you have the AAV responsive reporter cell line, which has a capacity of detecting the transduction of AAV vectors and then express luciferase. If this transduction is reduced or blunted by the presence of neutralizing antibodies, the production of firefly luciferase is going to be reduced or blunted. To run this assay, you first need to run the activation assay, in which we titrate the amount of packaging cells versus AAV responsive reporter cells. This experiment only needs to be run once, and it takes an overnight. Then you can run the neutralizing assay, in which you are mixing the cells together with your serum samples, with controls, and with the reference. In this case, as a reference, we use QVIC, which is IgG pulled from human donors. Then we can obtain this dose response curve that is going to allow us to determine the percentage of inhibition of each individual serum. This assay, again, only took one overnight to run. Let me show you some data. Here you can see the results of testing 20 different individual human samples for the presence of neutralizing antibodies. This experiment was run with a final concentration of serum of 10%. As you can see in your left-hand side, we use the QVIC to obtain a dose-response curve that allows us to determine the 0% inhibition and the 100% inhibition. This way, we can determine the different percentage of inhibition of each of the individual human serum. Then we selected 4 of these individual human serum. In this case, we select the pink one that represents a non-inhibitory human serum, the light blue, which presents a small inhibition capacity, the dark blue, which presents a high inhibition capacity, and the green sample that presents the highest inhibitory capacity, to be able to titrate it further on. In order to evaluate the inhibitory effect of each of these 4 individual serums, we perform different dilutions as shown here. We do the samples 1 to 10, that was the initial concentration we used in the determination of the presence of neutralizing antibodies. We also dilute 1 to 50, 1 to 500, etc. Then we repeat the same experiment to perform the percentage of inhibition in each of these points for each individual serum. We could observe, for example, that after 1 to 50 dilution, the sample number 3 already lose the inhibitory capacity. However, sample number 6, and especially sample 15, still have a high percentage of inhibition, suggesting that both of these samples have a higher titer against AAV2. Based on the data that I have shown you, we can conclude that the iLite AAV neutralizing platform can be used for screening sera for the presence of neutralizing antibodies, that this system is serotype specific. Right now I have shown you data detecting NAbs against AAV of serotype 2, but we are currently developing against serotype 5, 8, or 9. Moreover, as I told you, as the system is very modular, we can produce a custom solution for any specific hybrid or chimeric capsid. The systems eliminate the necessity of purifying or producing a new vector, and the cells are available in a frozen thorn-ready use format. So to conclude this talk, let me say that the development of bioassays for the cell and gene therapy field highly depend on the outcome we want to assess. First, if we want to develop a bioassay for a specific therapy, it is very important to mimic some characteristics, especially the cell type. In this case, in the iLite cyclic GMP, we were using the human retinal cells, and to try to reflect the mode of action. Again, in the case of the iLite cyclic GMP cells, we are using the cyclic GMP metabolism to mimic the effect of the vector. This type of assays can be then used for assessing the potency of the vector, improving vector optimization, and comparing, for example, manufacturing methods. But if we want to develop a bioassay for a more analytical application, it is very important to use a well-defined technology in which we can totally rely on. For example, in the development of a bioassay for determining the presence of neutralizing antibodies, it is very important to use a bioassay that is vector detached, which means that you don't need a specific AAV production or a specific AAV purification. Moreover, you need a bioassay that is very flexible, because you're going to use it in a different wide range of scenarios. In the case, for example, of the iLite AAV neutralizing platform, I have shown you the data using AAV2. But this same system can be customized for AAV5, AAV8, AAV9, etc. Because the system is very modular, so it can be set up and tailored for specific capsids. In the end, we would like to thank you for your attention, and if you have any comment or any question, you can contact us through email or via our webpage. Svar is much looking forward to also providing answers to challenges in life science. Thanks.